Well, thank you. Good morning to everyone. First of all, I would like to thank to all the attendees and overall speaker of today for their presence in this event. First, let me present myself. I'm Licinio Diaz. I'm head of the Industrial and Environmental Biotechnology Unit at the ITENE Research Center. We are RTO. We are located in Valencia, in Spain, and I have the honor to be the coordinator of one of the three projects that will be commented today. In that case, Encycon. The aim of this event is to highlight the potential of biotechnology for the recycling of conventional plastics in different sectors, such as packaging, agriculture, transport, automotive, construction, etc. And to this end, the EU Commission and overall the bio-based industry joint undertaking that is very well represented by Antonella Canalis, that also her is she's a project officer of two of these projects, in that case of Encycol and Recover. And this association have the aim to take the leap forward for implement this development in the European industry. During this event, which will last approximately more or less 3 hours, first of all, we will have an introduction from sorry, I have to move to the agenda. Yes, we will have an introduction from BBI that I said is very well represented by Antonella Canalis, project officer of Encycol and Recover, and she will talk about how BBI is promoting the bioeconomy through Europe and how this project will contribute to do it. Then we have 3 blocks, one from each of the project, and we will show a small overview of each project, and we will highlight the different development and how each of them meet and face the different challenges that we have in front of us. And finally, we will have a section of question and answer to answer all the questions that you may have or may appear, or suggestions or whatever. In that sense, in this time, we will try to solve and to discuss about the different projects, the different cooperations. And through the event, you can write your question in the chat and indicating to whom they are addressed. And in this time, at the time frame, we will answer them. Well, thank you very much to all of you. And right now, I leave the floor to Antonella Canalis from BBI.
Can you hear me?
Good yes, very well.
Very good, excellent. So, dear participants, speakers, good morning. It's a real pleasure also for me to join this event organized by the 3 projects within the frame of the European Green Week 2021. My name is Antonella Canalis, and I am a project officer at the Bio-Based Industries Joint Undertaking, the BBIGU. Indeed, plastics is an essential material in our modern life, but also has led to significant negative impacts, for example, causing harm and pollution in land and marine environments. Today, we are gathered here to talk about the development of innovative solutions by using enzymes to tackle end-of-life issues of plastic. We can all agree that this is a very fascinating research area. I would like to highlight 2 aspects. It contributes to addressing the dramatic problem of plastic waste, one of the major threats to our ecosystem, and secondly, it promotes circular approaches. It is in fact expected that the developed solutions are able to convert waste streams into new feedstock for the bio-based industries. 3 BBIGU projects have received funding to work on reducing the malign impact of plastic on the environment while exploring new business opportunities. Again, these projects are Bizente and Cycle and Recover, and very proud to be the project officer of 2 of them. In the following presentations, each project will tell us about the specific challenges and problems that they are addressing and the technical solution they are working on. Industries, university, and research and technology organizations working together within these 3 projects have the knowledge needed to develop solutions to the degradation of non-degradable polymers by applying bioprocessing-based approaches. BBIGU is very much looking forward to seeing the results that these 3 projects will generate. I would also like to add that these projects present here today, together with other projects funded by BBIGU, work towards achieving other objectives, such as ensuring that biodiversity is preserved and reducing the dependency on non-renewable resources. Europe has been recently called to achieve a very ambitious goal. The European Green Deal is a new growth strategy aiming at transforming Europe into a fair and sustainable society, with efficient use of resources by moving to a clean and circular economy. And last but not least, at making Europe climate neutral by 2050. Let me say that the good news is that BBIGU, the only policy initiative at the EU level on the bio-based industry, has already substantially supported the European Green Deal. For example, 64% of our BBIGU projects report that they address circularity and waste reduction. Yet, 58% of BBIGU projects report a reduction of greenhouse gas emissions along the new bio-based value chain. But there is further good news. The Green Deal considers mobilization of research, advancement of innovation in the circular bio-based sector critical to achieve its goals. Horizon Europe, the European framework program for research and innovation that just started this year, will support the needed research and innovation efforts. We have long-term goals for 2050. The real job has already started and needs to be continued today and the years to come. Let me tell you about a new initiative that is in the pipeline. We are not yet there, namely the Circular Bio-Based Europe Joint Undertaking, CBE. This is expected to be the successor of BBIGU. CBE would aim to promote a circular and sustainable bioeconomy, which is a core instrument for the Green Deal. The proposal for the CBE has been adopted by the Commission and it is still under discussion. I could not miss this opportunity to give you a short presentation of what BBIGU has achieved and why it is considered a successful initiative. I will share now my presentation. May I ask you if you see it?
Yes, Antonella, but we can see the secondary screen.
And then you see, pardon, Marta?
We can see it, but the secondary screen?
Let me see if I can.
Yes, we are seeing the presenter view, maybe in display setting on the top of the screen. On the top in display setting option in the presenter view.
I don't see it.
You have 3 menus on the top of the screen. Display setting, yes.
Yes, here you are.
Yes, that. And duplicate slideshow.
Okay, very good. Excellent, thank you. Do you
Right.
see it now?
You're welcome. Yes, perfect.
Perfect. So just want to tell you 2 words about BBIGU. BBIGU was established in 2014, and the objective was to support the development of sustainable and competitive bio-based industries in Europe, based certainly on advanced biorefineries that could use biomass in a sustainable way. How does BBIGU meet this objective? By implementing the Strategic Innovation and Research Agenda via call for proposals under Horizon 2020. The BBIGU is a public-private partnership funded by the European Commission from one side and the Bio-Based Industry Consortium from the other side. It has been established in order to de-risk investment in this sector, to reach critical mass, to mobilize actors and new sectors, and organize the value chain. These are the key figures of what BBIGU has delivered in terms of projects. 142 projects have been funded with an overall investment of 821 million. It is expected by 2024 that €1 of BBIGU has generated a contribution coming from the private sector of 2.8 euros. The initiative has mobilized more than 1,000 beneficiaries from 39 countries. These beneficiaries are unique beneficiaries, meaning that many of them are participating in more than one project. BBIGU realizes its objective via using 4 actions: research innovation action, demonstration, flagship, and coordination and support action. We have funded 71 RIAs aimed at developing new knowledge, new technologies to fill in the technological gaps which are in the value chain. We have funded 39 demonstration projects, demo build new plant, new demo scale plant aimed at demonstrating the feasibility from a technical and economic point of view of a new business model. And we have funded 14 flagship projects. The objective at the beginning of BBI was to have only 5 flagships. We have indeed, we did really beyond this target. Flagships are projects that aim at basically realizing biorefineries. They are very important because they allow the deployment of business models at industrial and commercial scale. And then the last action are the CSA, which aims at accelerate the market uptake of the bio-based product. This map shows you how the demonstration plants and the biorefineries are located in. It's clear that these projects are well spread over Europe. We have 14 flagships projects with high potential of being replicated, which are well spread from France to Spain to Ireland to Romania, etc. And then, yes, the type of organizations in our funded project. Overall, there is a broad participation of different entities, including academia and research organizations. There is certainly a solid mobilization of the private sector. BBIGU is an industry-led program with an overall participation in terms of unique participants of 66%. But the SME represents 41%. This is well beyond the target of Horizon 2020, which was set to be 20%. But it's very fundamental within BBIGU, the participation of universities and research organizations. In fact, although BBIGU is an industry-driven program, it's very much focused on the collaboration between private and public, between academia and industry. This in order to develop new technology to overcome obstacles and then allow the advancement of the bio-based industries in Europe. University and research organizations overall represent 26% of our participants and have received just a bit less than 1/3 of the BBIGU grant amount. In terms of feedstock, this graph shows what kind of feedstock is used by our BBIGU projects. We have agri-based feedstock, forest-based, organic waste, municipal solid waste, mainly aquatic and other feedstock. And this is the novelty. These three squares that you can see in this graph are exactly the three projects: Recover, Bizente, and Cycle. Certainly, the main source of feedstock appears to be agri-based, followed by forest-based. But nevertheless, increasing over time the use and the valorization of organic waste and aquatic. And in 2020, we have for the first time 2 flagships valorizing organic waste and aquatic, mainly algae biomass. From the feedstock to the final application, this shows where the bio-based sector and which final application is working on and which final products is delivering. The most frequent applications, certainly food ingredients, feed ingredients, packaging, but also interesting application in the textile sector, home care, and automotive. The mantra in BBIGU has been always unlock the full potential of the biomass, having the more from the less, having more than one product from the same biomass. This by applying cascading approach and integrate bioraffineries. BBIGU projects are not requested only to deliver bio-based products, bio-based building blocks or materials, but it's fundamental, and this is really a condition sine qua non for the proposal to be selected, that they deliver also on environmental impacts. The majority of our projects report that they are contributing to the development of the sustainable bio-based industry in Europe with a reduction of the negative environmental impact by certainly using circular approaches, waste reduction, recycling. They are contributing as well to the climate change mitigation, as 58% of our projects report a reduction of greenhouse gas emissions. But our projects are also delivering in terms of expected socioeconomic. 83% of our projects report that they are contributing to creating new jobs, especially in rural areas, 53%. And the impact on jobs is certainly important for flagship projects. And certainly, as already anticipated, the impact on science and knowledge. The strong collaboration between industry and academia and across regions and countries is creating knowledge that can be translated into new business opportunity, into new markets for the bio-based products. Let me set the scene for the 3 projects that have been funded by BBIGU in 2019. Want to share with you just some data. In 2018, 29 million tons of plastic were collected in Europe. 32% went for being recycled, 43% for incineration, and the remaining 25% for landfill. Indeed, the percentage for recycling is possibly much less because of the difficulties also of recycling plastic. Starting from this consideration, and the question mark for the bio-based industry was how can we also contribute to reduce the impacts on climate and on the environment. We started from two considerations. Plastic waste is a resource for the bio-based industry, and there are already bioprocessing that can be used in order to tackle end-of-life issues associated to the plastic. Now, these three projects are exactly addressing end-of-life issues of plastic. I will not tell you what they are about. What I'm interested to tell you that there is a strong complementarity between these three projects. All of them are addressing different problems related to the degradation of polymers, but all of them are also generating new feedstock that can be reused for the bio-based industry. So I'm really happy to have been invited here to this event, and now I thank you for the attention. And please, if you want to be updated with the news of BBIGU, please register at our BBIGU newsletter via our website. Thank you for your attention, and thank you for having me today. Wishing you all a fruitful meeting.
Thank you so much, Antonella, for your great presentation. I think that BBI is a powerful engine to boost all these technologies and to implementation to the industry. And thank you so much for trusting on these three projects. And we will try to do our best to achieve all the objective. Right now, let me share again my screen, and it's time for we will continue with the with this first block is regarding Encycle, one of the project granted by BBI 1 year ago, in order to try to boost this kind of technologies for recycling conventional plastics. As I introduced before, my name is Licinio Diaz. I'm the project coordinator of Encycle, and I belong to ITN Research Center that we are located in Valencia in Spain. Well, this is, I will, from Encycle part, I will speak at first time trying to introduce a little bit Encycle to all of you, and after me, Christian Sonnecker from the University of Leipzig and Aswin Reinhardt from the Indorama will explain different point of view of the Encycle project. One is more focused in the enzyme development on how we will promote this enzyme technology and the discovering of enzyme for this special application. And from Aswin from Indorama, we will have an industrial perspective and remark the relevancy of this development in industry and the boundaries and the advantages and disadvantages maybe that this kind of processes have. Okay, regarding Encycle, I will speak about a little bit on it. This is only a few tips about this project. If any of you have additional questions and so on, in the time of question and answer, we are very glad to answer all your questions, and by the different channels we will communicate with all of us. Well, as I commented before, this project is coordinated by us, by ITN, have a duration of 48 months, from more or less from the 1st of June in 2020 until May of 2024. We have a budget a little bit of 6 million euros, and we count with an in-kind contribution for the different companies, almost 2 million euros. Okay, regarding the consortium that perform this project, we have 13 partners. We have different FTOs, SMEs, and 5 large companies in which we are all cooperating between all of us to try to achieve this objective. A brief sum up, we count with Asa Special Enzymes, is an enzyme producer located in Germany, and also with the University of Leipzig. That is one of the, I think, that the biggest experts in this kind of technologies. From Netherlands, we count with Indorama, with Feyecon, expert in supercritical technologies, and Wageningen, that is a very known university. From France, we count with Soprema in order to validate different monomers that we want to obtain in order to promote different construction materials like polyurethanes. From Austria, we count with Rainer, with ASIP, and the University of Vienna, BOKU, in different aspects for enzyme production, of course, communication and dissemination in this project, and also the degradation of different kind of polyolefins. We count with Aliplast. In Italy, this is they belong to the group ERA, and it's a big waste manager and recycler in Europe. And finally, from Spain, we count with DAM, is the acronym of Depuración de Aguas del Mediterráneo, more regarding with the wastewater treatment. And Darwin is a SME, more focused in development of new microorganisms with special capacities or special metabolic capacities for that. And finally, ITN, we are a research center more specialized in packaging, transport, and logistics. And in that sense, we participate in this project not only as coordinator, but developing the new processes, new enzymatic, the polymerization processes, and so on. Well, this is in this slide, you could see the main problem that we have. I am aware that many of you are aware regarding the problems that the Europe has regarding the plastic management or the plastic waste management have. In that case, in this project, we will focus in packaging and also in microplastics, and there are a few numbers regarding the huge economical and environmental impact that this fraction represent to the society. This project, Encycle, aim to trying to develop new processes based on new enzymes to recycle non-valorized plastics fraction, and in that sense we will deal with 3 different targets. The multi-layer packaging that, as we know, they are not recycled today due to confusion in the sorting plants and in the waste management plants. More focus in this kind of packaging, we will focus in the multi-layer based on polyethylene and PET. After that, we want to deal also with post-consumer polyethylene terephthalate trays and cans and containers. And finally, with microplastic present in wastewater. We thought that this technology could be very valuable for this target and to implement in this kind of industries. And for this reason, we include in this project. Overall, the problem it represents is quite important to deal with that. And well, this is the overview Encycle in a picture, in which in this project, the overall objective is to develop new enzymes and advanced processes for the enzymatic treatment of non-recycled plastic fraction. In that case, our idea is to produce updated, upgraded new material such as virgin PET, polyurethane foams, and polyols, and reduce the environmental impact generated by microplastics. In this project, we will cover the whole value chain in not only the enzyme production, but also the process development. We have different steps, and this project starts with the microorganisms, with these special metabolic microorganisms, which are able to degrade different plastic fraction. In that sense, in this slide, you could see the expanded flow diagram. In that sense, in this part, this is all related with the development of these new strains. We will select the different enzymes and we will clone, and also we will try to perform innovative technology for the production of these enzymes. I think that is a key point in this project. In that sense, we count with these three targets that we commented before. And for the first target, we will try to implement different technologies, different pretreatment, and also different enzymatic processes for the recycling of this multi-layer packaging. In that sense, we will deal specifically with the polyolefin layer and the PET layer. Regarding the PET, clamshell or the PET trays, this is some kind of similar flow diagram in case of multi-layer packaging, but in that case we will focus with PET. These monomers will be used for the polyurethane synthesis and the PET repolymerization in order to obtain a proof of concept. This is more or less the idea. And in case of microplastic, we will deal with three processes that currently is taking place in the waste management plant, in the wastewater management plant. Sorry. In that case, we will try to develop a new system for the degradation of these micro nanoplastics based on enzyme mobilization, and also we want to implement this system, the system of the implementation of these enzymes in process like in traditional biosensation processes such as anaerobic digestion and composting processes, in order to avoid the generation of microplastic and the spreading in the environment. Well, from my side, this is a very brief overview of the project. Please stay tuned to this project. We have the website. We are very active in social networks like Facebook, Twitter, LinkedIn, and we keep at your and we are available for all of you and trying to talk about this project of some of our development. Right now I will leave the floor to Christian Sonneke from the University of Leipzig, and please, the floor is yours.
Christian, we cannot hear you. I'm not sure if you're talking.
Yeah, do you hear me?
Now, yes.
Do you see the screen? Do you see a moon?
We did, but now you stop sharing.
Okay. Let's try it again.
Christian, you are not.
We cannot either see.
Okay. I have it's sorry. I try
Now
again.
it's fine.
Yes, right.
Okay, now you see the moon?
Yeah.
Okay,
yes.
let's try it again. If it doesn't work right now, then just tell me. I try it again.
Yes. Right.
We can see it now. But for whatever reason, we cannot hear you. We can see it, but we cannot hear you.
Okay, I got it. Okay, sorry. Sorry for the trouble. Yeah, I have to take another slide. Sorry, 1 second. Okay. Now you see the screen?
Yes.
And now you hear me?
Yes.
Okay,
Yes.
great. Okay, so I'm okay now. It's working. Okay, yeah. Thank you, Lisinio, for the kind introduction. So now I want to tell you how we will try to obtain the corresponding enzymes, which we will then use for the biodegradation of PET materials. So of course, so we are searching new enzymes in the environment. So therefore, we have to go out into the environment and pick the samples. And so we are taking samples from a total of 50 different spots. And after collecting the samples, we will extract the metagenomic DNA from the samples. And that has one big advantage, because if we want to have a good overview about the microbial community, it is very hard to cultivate these communities, because maybe only 1% of the bacteria in these samples might be culturable in the lab. That means we would lose most of the information. However, when extracting the metagenomic DNA, we can also get access to the information we could not access by cultivating the strains. So therefore, this has a big advantage for us. And when we have isolated the metagenomic DNA, we are then performing a fragmentation of the DNA. So of course, for each sample separately, and then performing next generation shotgun sequencing. We are thereby obtaining small reads, which we are then forming longer contigs with in silico methods. And from this information, we can get an overview about the microbial composition of the sample. And that is very interesting for us, because we already know promising candidates harboring the desired enzymes. For instance, these are actinobacteria. And now when we here we see the microbial composition of one of the samples. When we have a closer look into the actinobacteria, we see there are a lot of different genera from the actinomycetes, and this is quite a promising outcome, because here we see candidates where we know that they are harboring such desired enzymes. So therefore, these samples are quite promising to search for polyester degrading enzymes. However, when we only obtain this information, we will not have the genes in hand. Of course, we want to have the genes to test them and then to optimize them. So how do we obtain the genes? I will show you now. So, so when we have the metagenomes in hand, and we are then performing a polymerase chain reaction using degenerate primers. And now you have to know that the genes encoding for polyester degrading enzymes, they are relatively conserved at their flanking ends. That gives us the opportunity to design degenerate primers that are binding to multiple sites. So we then can fish out more or less a variety of genes. And then after we obtain the amplicons from the PCR, we now have to test whether the genetic or the DNA contains some useful information, namely encoding the desired genes. So what we plan to do then is to perform amplicon next generation sequencing. And this will give us an overview about the sample composition. So how much different sequences do we find? Do we find the polyester degrading enzymes, or is it some other DNA fragments. On the other hand, of course, we have to test the fragments if they are active against polyesters. So therefore, we are cloning the amplicons into an expression vector. And we are screening in an E. coli platform or E. coli screening system against polycaprolactone and polyethylene terephthalate agar plates, and clones that are positive will show a halo. And with these clones, we can then use these clones to make a further or deeper characterization. So when we see the halos, we can then take the clones performing a plasmid extraction and make a Sanger sequencing. So now we have the gene information in hand, which will already give us a good estimation whether or not these genes are promising candidates for polyester degrading activity. So when we then, of course, have to test the enzymes by expressing and purifying the enzymes to determine their characteristics. And cycle in a first screening round, we will use the E. coli platform. However, we also plan to scale up the system in the later phase of the project. So therefore, we will also test different prokaryotic and eukaryotic expression systems to increase the yield of the enzyme, because this is also an important factor when you want to make a scale up. So most importantly, we are interested in the thermostability of the enzymes, because we know that especially when you want to degrade PET, you need a certain reaction temperature near the glass transition temperature of the PET material, because there the PET chains are getting more flexible and the enzymes can then process the hydrolysis more efficiently. So when we know about the thermostability, we will also have a look towards the substrate specificity to see whether the enzymes are more active towards longer or shorter substrates. Then we are checking the general PET degrading activity. And of course, we also want to test the PET film degradation. And that's of course important for us when we want to degrade complete PET films. We want to know how much time it will take. And of course, we want to reduce the time as much as possible, as of course this is also a critical factor when you want to make an economic process. Of course, time is money, so therefore it's good to have the so now at the second phase we will or later we will then make the protein engineering of the most promising candidates. Therefore, we will use computational analysis. So that means we are checking the amino acid sequence. And from that amino acid sequences, we can already get some ideas about mutational hotspots. Then we will also use structural information and in silico approaches like molecular docking and MD simulation to optimize the enzymes. And then when we have the best candidate, we will then test the candidate to degrade real post-consumer PET. And therefore, we focus on rather amorphous PET materials like clamshell tray packaging, because only these materials are directly biodegradable by the enzyme. So having higher crystalline PET materials, like for instance PET bottles, they will not be degradable by the enzyme. So therefore, we need pretreatments to make the chains accessible for enzymatic hydrolysis. So with these words, I'm ending my part of the session. And now Ashwin from Indorama will proceed, give a short overview about the next part of the presentation. Thank you for your attention.
Thank you, Christian, so much for your great presentation. Now I will. It's the time for Ashwin Reinhardt from Indorama. Ashwin, the floor is yours.
Thank you, Lucinio. Let me
You're welcome.
check the presentation. Okay.
Okay, that's it. Perfect.
Okay. Thank you, Lucinio. Thank you, Christian. Good morning, everybody. A quick introduction about myself. My name is Ashwin Brijlal, and I'm currently working as a development engineer within Indorama Netherlands. I'm personally based in Rotterdam, and to give you some more background information regarding Indorama, we are one of the largest global producers of polyethylene terephthalate, also shorted PET. And yeah, as you are familiar with PET, PET is globally used in several applications. It's used in the textile industry, but here at the location in Rotterdam, we are producing PET that is produced for several food packaging applications. You may think of bottles, trays, and PET film sheets. And yeah, as of today, Indorama is a partner within the Enzycon Consortium, and I was asked to share the industrial vision of Indorama regarding the technology of enzymatic recycling. So I prepared some slides, and yeah, if there are some questions, I think that at the end of this webinar we have a Q&A session, so we can. Oh, no, can you see the slide? Hello?
Yes, we can.
Oh, okay. Now my monitor suddenly went to sleep mode.
Oh.
So, please give me a minute to. Yes, okay. Sorry for the inconvenience. Yeah, so if there are any questions, we have a Q&A session at the end of this webinar. So the topics that I would like to discuss in today's session are yeah, a brief explanation regarding the post-consumer recycling system in the Netherlands, the current situation regarding global plastics recycling, yeah, an assessment and development of various recycling technologies that are currently available and their respective environmental impact. And finally, the added value of enzymatic recycling. Okay, so to give you all a brief overview of the post-consumer recycling system in the Netherlands. At the moment, yeah, PET bottles are collected either through a deposit return system or through separate collection and sorting systems using municipal waste streams. The PET bottles are then pressed in these so-called bales and then subsequently transported to a recycler. What the recycler then do is they sort out the PET, they wash it and they shred it to produce the PET flakes, which you can see in the picture. And depending on the color of the flakes can end up in various applications. So, for example, the colored flakes will end up in strapping or fiber products. The colorless flakes will end up in PET trays, and the color flakes can also be used to make the so-called rPET pellets, which then can be used to make PET bottles out of it again. And based on this system, we can see that the input for the system is of course heavily relying on using really clean input streams in large quantities, and also to make use of optimized sorting and cleaning technologies in order to produce these high quality PET flakes and pellets. So having discussed the recycling system of PET bottles in the Netherlands, I would like to take you to some recycling numbers on a somewhat global level. So in 2018, McKinsey and Company conducted a study regarding plastics recycling worldwide, and their research showed that only 12% of all plastic waste is currently being reused or recycled. And as you can see in this chart, the great majority, 84% of all the produced plastics, either goes to incineration, landfill, or is being dumped or leaked into the environment. And well, the amount of plastics that cannot be captured is enormous. And yeah, we believe that the greatest challenge is to make sure that we are able to efficiently utilize these polymer waste streams. So if we look at the concept of recycling, we know that several levels of recycling categories are distinguished. So the first one, we have non-circular pathways. This is basically an example could be incineration to recover the energy content in the plastic, but by incineration you will lose the product, so it will not be available anymore for any reprocessing needs. Then we have, of course, mechanical recycling. It's a process where the polymers are remelted and reprocessed into new products, but remelting the polymer can negatively affect the quality of your final product, but the recyclers are able to blend this product with virgin material to still obtain good products. Another form of recycling is gasification and pyrolysis, where the plastic waste is downgraded to produce syngas or other liquid products, such as pyrolysis oil, to produce, for example, fuels and chemicals. But the final form of recycling, where I want to focus on, is the process of depolymerization, where the polymer is chemically reduced to its original monomer form, so that eventually it can be repolymerized. So in 2018, a study was published by a Dutch think tank company called CE Delft. And in this study, the research found that chemical recycling technologies in the Netherlands are better suited for treating mixed plastics waste streams that currently cannot be processed through mechanical recycling. And another observation in the study was that if you look at the technology of depolymerization, in this chart you can see that it will save 1.5 ton of CO2 per metric ton of PET trace waste processed. Mechanical recycling will give you an environmental benefit of 2.3 tons of CO2 per metric ton of PET trace waste processed. But in the end, mechanical and depolymerization technologies are far much better when compared to incineration, looking from an environmental perspective. So all in all, we can conclude that chemical recycling can be considered as an interesting technology to complement today's limitations of mechanical recycling. So within the field of chemical recycling, we noticed that the concept of enzymatic recycling has gotten more and more attention nowadays. And we believe that this technology may provide an efficient and sustainable way for managing PET waste streams. So, yeah, we know that scientific studies have shown that enzymes are considered to be highly specific to target a suitable molecule. So in this case, it would be PET. And heavy colored mixed plastic waste streams containing a variety of other plastics such as PE, PP, PVC, which currently cannot be processed by mechanical recycling and are ending up in landfill or being incinerated, could now be utilized by the usage of enzymatic recycling. The products that we will obtain after enzymatic recycling, PTA and EGA, can then be, for example, easily be repolymerized by Indorama. We already make use of these building blocks that are fossil-based, and using these building blocks, we produce PET. But using enzymatic hydrolysis, we can break down the PET into PTA and EG, which then can be repolymerized by Indorama into PET. And then basically, we are able to create an infinite process of production and recycling, and to also decrease our dependence on fossil-based raw materials. So in the end, we believe that the combined effort of all partners within the Encycle Consortium will make a significant impact in the field of enzymatic plastics recycling, and that together we can contribute to a more sustainable and circular economy. I'd like to thank you all for your time, and yeah, if there are any questions, I would be more than happy to answer them in the Q&A session.
Thank you so much, Ashwin, for your great presentation and the implementation of the relevancy of the implementation in the industry. Now I think that we finished the block related cycle project, and we move on for the next one, that is Vicente. In that case, coordinated by ITIP and other Spanish research center located in Zaragoza, and thank you all for the hosted of this event. Now, Marta.
Thank you, Licinio. Good morning, everyone. Thank you for your time and your interest in the three projects. As Licinio said, my name is Marta Redrado. I work at ITIP, and I'm the project coordinator of Vicente. How we are going to do our session is I'm going to give you an overview of Vicente, and after that some of our partners will explain you a bit more in detail what we do. So let me start sharing a video with you that I hope it works. Let's see.
Composites plastics are present in everyday elements, such as airplanes, trains, windmills, buildings, vehicles, electronics, or sports equipment.
No sound.
Okay, sorry, let me try.
They allow us to improve our lives in many aspects, thanks to their greater stiffness and lightness. However, there is no final solution for the valorization and reusability of thermoset composites materials. Once they reach their end of life, they are dumped in landfills, awaiting measures to enabling their reuse or incineration. It is forecast that 430,000 tons of composite waste will be produced worldwide in the current year, which will have a highly negative impact on the environment.
Or we can.
Okay.
Vicente project, which involves 10 European partners, rises to the sustainability and circular economy challenges that the European Union faces. By breaking down at least 27.5% of the total thermoset composites waste, this enzyme innovative technology will also contribute to a 40% or greater reduction of non-biodegradable thermoset composites emissions into the environment, thanks to an efficient and sustainable process.
After this very brief video, let me give you a brief introduction. As mentioned, Vicente is a European research and development project which is being funded by the European Commission within the Horizon 2020 initiative and also by BBIGU, the Bio-Based Industries Joint Undertaking programs. This project involves 10 partners from 5 different countries, you can see them there in the map, and 50 expert professionals from a very wide range of fields. Plastic waste is one of the main threats to our ecosystems. Currently, there is no final solution for the valorization or reusability of thermoset composite materials. At the moment, what happens is that they are dumped in landfills or they are incinerated. Both solutions or both alternatives mean a harmful footprint on the environment and public health. So what Vicente proposes, the target of the project, is to develop a new technology which is based on the use of mutated enzymes to carry out the degradation of these thermoset composites. These thermoset composites are based on epoxy, polyester, and vinyl ester resins, and what we are trying to do is to resolve these, the current end-of-life issues we have. This technology will be mainly studied in plastic residues from aviation, transport, and construction. After this really brief introduction, and in order to understand better Vicente, let's start explaining what a thermoset composite is and how it is fabricated. For that, Julio Vidal from ITIP is going to give us a very brief explanation. Julio has a degree in chemistry and a master in nanomaterials. He has been involved in many competitive funded projects at national and international level. He has knowledge in the definition of new formulations for advanced materials, in which he is working for his PhD. Julio, the floor is yours.
Hello everyone, my name is Julio Vidal. I'm from ITIP, in the Technological Center in Zaragoza, in Spain. And I'm going to present to you thermoset material and how we manufacture them. I think that we all know what's a thermoset material. It's a material that when you heat it up, it doesn't melt, it degrades before melting, so you cannot melt it. So I want to focus my presentation really short in just, I hope it will be just 2 minutes, in how do we manufacture thermoset materials and how we work with thermoset materials. As you can see in here, in order to manufacture a thermoset material, we need a mold and it has to be properly cleaned. For the edges, we use acetone in order to allow the thermosets when it's formed to be not to be attached in these sides of the mold, to keep them cleaned and proper. And in order to demold the material, we have to use a decoupling agent inside the mold. Okay, that will allow us to demold, to take out the final piece of the mold easily. The next step in the manufacturing process is to place the different reinforcement materials inside the mold. In this case, we use 3 different layers of non-woven material, and to place some absorbent around the reinforcement materials, just to take all the extent of matrix that we are incorporating into the material. This will allow us to not have more matrix than we need in the final composite. Once we have properly placed the reinforcement material, we have to use different plastic layers to allow a proper distribution of material, of resin, of matrix inside the mold, inside the final demonstrator. In order to take out any air bubbles that might form during the process, we are going to do some vacuum inside the final demonstrator that will allow us to avoid any defects. Okay, this vacuum is also used to pump the resin, the matrix, from out of the mold to inside the mold. In there, it will impregnate all the fibers, and it will cure as the mold is with the necessary temperature. It might depend. The temperature might depend on the on the resin that we are using. So we will finally, after some time of curing, we will finally obtain a piece as you can see on your screens. So finally, once we have cured the material at certain temperatures, as I mentioned, that depends on the kind of resin that we are going to use, we will take out the material easily because we have used this decoupling agent cleaning the mold. We will take out all the different plastic layers, and we will obtain a final demonstrators, as we are using LRA infusion, liquid resin infusion. What we will have is a really a well finished phase, and another phase that will be not so well finished. So in this case, we will just have one aesthetic phase by using this technique. So thank you very much to all of you. I hope it was quite easy to understand, that you have no doubts. But in case you have, please do not hesitate to contact me either by phone or by email. I will be glad to answer all your questions, and have a nice event.
Thank you. Thanks a lot, Julio, for your presentation. Now let's see what is the methodology that we follow at Vicente. We are going to start with Dr. Javier Viña. He's the co-founder of EvoEnzyme. He has a degree in biochemistry and is specialized in direct evolution of enzymes. He's going to explain us what we do in Vicente with the enzymes. Please, Javier, the floor is yours.
Good morning, everyone. My name is Javier Viña, and I want to introduce briefly the importance of enzyme engineering in biotechnological applications. Biocatalysis is one of the main fields in development to build up greener and eco-friendly processes for nowadays industries. Enzymes are very selective and efficient catalysts. However, to be applied in industrial settings, they should be adapted in terms of high yield production, stability, or catalytic performance. One of these large-scale applications, and very relevant in the European Green Week, is the biocatalytic degradation of plastics. Within that framework, Vicente project will develop a biotechnological solution to the end-of-life issue of thermoset composites. In the laboratory, enzymes are modified and adapted by means of genetic engineering techniques. Probably the most powerful methodology presented today is directed evolution. Based on the algorithm of Charles Darwin with diversity generation, selective pressure and evolution, Professor Frances Arnold from Caltech developed a protein engineering technique for which she won the Chemistry Nobel Prize in 2018. Natural evolution is a spontaneous process of millions of years. Directed evolution, where mutated variants of enzymes are created by point mutation and DNA recombination, is a process totally controlled by the scientists and takes only months of bench work in the laboratory. The lignolytic consortium secreted by fungi with enzymes like lactases and peroxidases is responsible for natural wood degradation. This enzymatic group is considered one of the most powerful oxidative systems in nature. Indeed, these enzymes are specialized in degrading a chemically complex and recalcitrant polymer as lignin. This achievement makes them the best candidates for degradation studies of complex plastics like composites. In the laboratory, mutated variants of these enzymes are expressed in Saccharomyces cerevisiae in 96 well-plates format. These microfermentations are studied in terms of activity or stability with high-throughput screening assays. In Vicente, several resins are being studied with different chemical nature like epoxy, vinyl ester, or polyester resins. For the directed evolution process, model molecules of these resins are selected as structural scaffolds. Mutated variants of the ligninases will be studied with custom-made colorimetric high-throughput screenings in the search of the most.
Thank you, Javier, for your presentation. Continuing now with Dr. Alain Graillot, he's going to explain us the resin modifications. Alain graduated in chemistry and engineering school in 2010 and later obtained his PhD in polymer chemistry. He joined a Specific Polymers team in 2013 as a research engineer, and today Alain is the Chief Operating Officer of the company and leads the industrial and collaborative R&D projects. Alain, the floor is yours.
Good afternoon. My name is Alain Graillot. I'm the CEO of Specific Polymers, and today I'm going to speak about the work we are doing in the scope of Bison project, which is dedicated to the resin modification of these thermoset materials in order to favor the biodegradation of these resins. So in the scope of Bison project, and more generally speaking within the company, we are really making research and innovation in order to solve out the end-of-life issue of thermoset materials. Indeed, these materials are not recyclable, and so we target to make chemical modification in these materials in order to make their recyclability available. So in the scope of the project, we wanted to focus on various kinds of materials which can be representative of all the materials that you can find in your life. And thus we focused on 3 different application fields: aeronautic, construction building, and railway. And for each application field, we defined reference resins which are currently used in this application. And this reference resin will be will give us the specification of the material we want to reach in order to have the same specification in terms of implementation, the same specification in terms of application, mechanical property, thermal properties. But what we want to do is to include in the materials reversible bonds that can be break at the end of life of the material. So the first step of the project and of our work in the project was to really define the main specification of this reference resin, to characterize them in order to define this specification. So we did it for both the epoxy resins that are used either in the field of aeronautic or in the construction building. We did it also for the vinyl ester resin, which is used by Acciona in the construction building, or for the unsaturated polyester resin, which is used for railway industry. When this specification defined, the second step was to make the chemical modification of the resin composition in order to favor biodegradation. And based on the state of the art we achieved on this subject, we identified 3 main ways to favor this biodegradation. First of all, thermoset materials are hardly degradable by enzymatic degradation because they are cross-linked materials, so the enzymes cannot go into the material. So the first idea is to break down the cross-linked network in order to recover a solubility or a partial solubility, thus to give access to the enzymes to the materials to degrade. And to do so, our strategy is based on Vitrimer technologies, in which we insert reversible bonds into the materials that can be break in certain conditions of temperature or solvents, and thus we can recover solubility. Second strategy, in order to increase the enzymatic biodegradation of the material, is to insert in the materials biodegradable functions. So for instance, it is well known in the literature that ester bonds or urethane bonds can be broken by enzymatic degradation. So our objective is to try to enhance the amount of such esterification or urethane moieties. And finally, because the enzymes are more effective in In water-like systems, the last strategy is to increase the hydrophilicity of the thermoset networks so the enzyme can work properly. So this is our three main technology we want to insert into the Bisant resins, and the first one is based on Vitrimer technology, and I would say the most important, recovers somehow some solubility at the end of life in order to give access to the enzymes. So just to show you what occurs when you do this kind of modification, so it's what we are trying to show on this slide, and we present the two different materials. I would say, can be representative of a reference resin. Here you have a normal hardener that is used, which is not reversible, and here we use a disulfide containing a hardener, which is reversible by disulfide metathesis. And so we prepared these materials, and then we studied their degradation in some specific condition of solvents. And what you can see on this slide is, thanks to the disulfide bonds, we succeeded to dissolve the thermoset materials, even if it's cross-linked at the beginning. And this is due to these reversible bonds. So it's this kind of technology we're going to apply for each reference resins, and this degradation will allow to achieve the enzymatic degradation at the end. Thank you.
Thank you, Alain. So now, once we have the enzymes and the resins modified, what is the next step? Professor Frank Hollmann is going to explain us. Frank studied chemistry in Germany, and he received his PhD in bioelectrocatalysis. He joined the biocatalysis group of TU Delft, the University of Delft, in 2008, where he now holds a professorship in biocatalysis. His research is focused on the use of enzymes for organic synthesis. Please, Frank, the floor is yours.
Welcome to this very brief introduction into the biocatalysis part of the Vicente project. In this part of the project, we focus on the application of polymer degrading enzymes, not so much on the real polymers themselves, but rather on low molecular weight model compounds. Obviously, now many of you will ask the question, why are they first using model compounds and not directly the real starting materials, the real polymers? Well, the answer for this can maybe be seen from this slide, because the real polymer products that we are going to deal with in the end are solid materials. And in a worst case scenario, the enzymes, the wild type enzymes or some of the engineered enzymes will not show any depolymerization activity on these sorts of materials. And then obviously we would be faced with the question, is that because the enzymes, the polymer degrading enzymes, are intrinsically not able to oxidize the polymers, or is it because the enzymes cannot sufficiently diffuse into the polymer products and thereby have not sufficient contact with the polymers? And that may be the reason for the absent or insufficient depolymerization reaction. That's why we first focus on low molecular weight compounds, because there we can at least see if the enzymes, if the ligninases that we use are principally able to oxidize and depolymerize the polymers. So by looking at the chemical structures of some representative polymers that we are going to deal with, it becomes evident that a common structural element of many of these products are glycerol and its derivatives. So this is a key core structure that is found in many of the polymers that we are looking at. Why we base our model compounds on glycerol-derived products, so such as these triethers of glycerol or amino derivatives of glycerols, which are still alkylated. So these are the model compounds that we are first looking at. What we are hoping for is to identify enzymes that are able to oxidize either this carbon atom or that carbon atom of the core structure. Because then, if we do get oxidation in these positions, we would also form reactive intermediates, which would spontaneously hydrolyze, which would spontaneously cleave the carbon-oxygen bond, which in the end would be a depolymerization reaction. And analogously, obviously, the same here in case of the nitrogen derivatives of glycerol. So amongst the many enzymes that are potentially able to oxidatively depolymerize these structures, we are first looking at two classes, being the vanadium-dependent chloroperoxidases and the unspecific peroxygenases. And using these enzymes, we basically follow two approaches. One approach is to have a direct interaction of the model compound and then later on of the real product, the real polymer, with the enzyme active site, and to have a direct oxygen transfer, for example, to this position. And the second strategy that we follow is to use these enzymes, to use the peroxygenases and the chloroperoxidases to form small, highly reactive oxidants, which then could hopefully diffuse into the polymer products and do the oxidation and initiate the depolymerization reaction. And in particular, when talking about highly reactive oxidants, we are mostly thinking about singlet oxygen species to be generated or hypohalous acids as very reactive compounds, oxidizing compounds, to diffuse into the into the polymers. So now I'm going to give you a brief overview over the results obtained so far. So we have synthesized these model compounds, and we have submitted them so far, for example, to the unspecific peroxygenase from Agrosciber Greta. And basically what we saw was that over time the substrate peak decreases and one or two or several product peaks actually accumulate. Right now we are not 100% sure about the chemical identity of the products, but we are working on the identification, and we actually already have a couple of ideas what these products could look like. And then obviously we use this information to our partners in the protein engineering part of the project to modify the selectivity of these enzymes. So overall, using this peroxygenase, we already have some promising results with at least one or two of the model compounds where we can see full degradation within a couple of minutes, actually in 3 hours in this particular example. In contrast to the results obtained with unspecific peroxygenase, the approach using this vanadium-dependent chloroperoxidase, where we base the reactivity on small reactive molecules, these results are not so promising at the moment, at least. We do see conversion of the starting material, which can also be seen in this chromatogram. We do see the formation of a product peak, but the activities and the conversions, at the moment at least, are not as high as we would like them to be. We have a couple of ideas what is going wrong at the moment. We are working on the reaction conditions, and then obviously again we will feed back this information to our protein engineering partners. So to sum up, the strategy of the biocatalysis part is to eventually use polymer degrading enzymes to depolymerize polymers such as this araldite polymer that is found here. But in order to screen enzymes that are principally able to depolymerize these products, we are focusing on these model compounds, establishing the activity and the selectivity of the enzymes with these model compounds, and then to further develop these enzymes further for higher activity, for better selectivity, for better depolymerization activity. Thank you very much.
Thank you, Frank, for your presentation. With this, we finished the block of the methodologies used in Vicente. And now to wrap up the session related to our project, let me explain you where we can find the materials we've been talking about. And also let me give you some figures and so you get an idea of what we're talking, and also I'll explain you a little bit the impact that Vicente is going to have. Thermoset composites were created in the 60s for the aeronautical sector. But as you can see in the graph here in this slide, since that time they've been used more and more every time. The objective of using these materials is to decrease the weight of the airplanes or the aircrafts and improve properties. That's why these materials are so interesting and so important for this sector. So you get an idea of how much composite materials are used in this sector. Here you can see a comparison of two different planes from two different providers. These are model planes, and as you can see, these have at least 50% of the total material is composite. So you can imagine the amount of waste we are going to have, and that's why we need a solution for it. At the moment, at the Teruel Airport, these two companies, Airbus and Tarmac Aerosafe, are dismantling planes and keeping thermoset materials in storage. These are some pictures of how it is now, and again, like you can see the amount of waste we are talking about. Another sector that uses a lot of thermoset composites is the construction one. Here are some examples that have been built by our partner Acciona. They are used, as you can see on the left of the slide, they are used in road and pedestrian bridges. And on the right, you have some examples that they are also used in marine works and structures which are built in harsh environments. Some of these pictures, you can see a lighthouse and also tunnels. As mentioned many times during our presentations, the problem of all these materials comes at the end of the life of these materials. Acciona has calculated that they have around 100 tons, a bit more than 100 tons a year of waste from this type of materials. This waste comes either from faulty productions or because the structures have reached their end of life and need to be changed. So the question is, what do we do with all this? What Vicente proposes, the solution that we propose, it's to apply enzymatic degradation of thermoset composites. This is really a pioneer idea because it's the first time that it's going to be addressed for this type of materials. The objective is to decrease the amount of non-biodegradable polymers that are currently discharged to the environment or sent to landfill and incineration by at least 40%. The estimations we have done is that Vicente technology will be able to treat at least 27% of total thermoset composite wastes. Another estimation we've done is that the project thinks we can do an extra reduction of at least 22% in the CO2 emissions due to the reduction in dependency of oil-based products. And also, another estimation is a cost saving of 10% thanks to use valorized products. The project also will have an impact on society. It aims at creating an effective after use of plastic, an effective economy of after use plastic. It will have social impact in rural regions such as Teruel, and it will help to develop high qualified skilled jobs related to the bioeconomy through, for example, our training plan. We expect to create 16 direct jobs and about 1,800 indirect jobs with an average growth for all Vicente partners of 13%. Well, and until here, an overview of Vicente, what is our aim and how we are going to achieve it. I hope it's a bit more clear now. I would like to thank you for your attention, and if you have any questions, you can use the questions and answers tab enabled during this session, or you can also drop me an email at any time, and if I cannot answer your question, I can direct it to the partner. So thanks again. So until here, the presentation of Vicente. There has been a small jump on Javier's presentation, but we are going to share the whole session with you once it finishes. So we will make sure his whole presentation is there for all of you who are interested. Now we are a bit delayed. We are about 20 minutes behind schedule. So we are going to have a little 15-minute break. So let's come back here at 5 to 11. And also I would like to tell you, we are going to start a polling questionnaire for you to answer. You'll have all the time you need until the end of the session. But we will really appreciate you can answer those really few questions that will help us improve for the next events. So thank you everyone again, and we'll see you back at 5 to 11.
Marta, are you there?
I'm here.
Can I try to share the video?
Yeah, let me just give you the rights. Hang on. Yeah, I'm gonna give you the presenter role. So in order to hear it, you'll need to share file. Okay, when you share a file.
Okay, thank you.
Now you should be able to do it.
Do you see it?
Yes, I can see it.
Okay.
Let's see if we can hear it.
Of the University of Almería.
Yes, yes.
And
okay, perfect. And if I pause it, and okay, perfect. It works, right?
Yeah, yeah.
Okay, thank you.
Perfect, thank you. We'll see you in 10 minutes, back in 10 minutes or so.
Perfect.
Can you see, sorry, can you see the poll?
Yes, I can.
Yes,
and
can you answer it?
Of course,
or,
I will.
Yeah, no, like, because I can't, as a host, I can't. So we just want to make sure there's other people who can do it. It's the first time we do this type of poll, so.
Perfect. I'll answer right now.
Okay, so you can do it. Perfect. Thank you.
No, wait. Now that I am let's say, I have the control, I cannot answer.
Okay, let me remove the control again. Yeah, that may now. Maybe because you are a panelist, you can't. Only the people who are not panelists can do it. That's
no, still I can't.
Okay, it's because you are a panelist, but that's fine. We'll just let the people, the attendees to reply. That's fine. Thank you. Go and have a coffee and I'll see you in 10 minutes.
Okay.
Thank you. Well, welcome back everyone. I hope you had a nice coffee, even if it was short. Now the project Recover is going to be presented. Rafaele is going to do the introduction to the project. So Rafael, the floor is yours.
Thank you, Marta. So welcome back everyone. And in this last part of our partner event, we will be watching a video presentation made by our Recover partners. So I would like just to introduce myself. I'm Raffaele Turri, working at ENCO, and we are involved in the communication, dissemination, exploitation activities of Recover. So I would like to introduce also Recover related, how is it related with the EU Green Week partner event? Because as you might know, this year the topic of the EU Green Week is the zero pollution ambition. And in Recover, we will contribute to decrease the microplastics arising from the agriculture and from the food sector. So with zero soil ambition. So our presentation will start with our project coordinator, Maria Jose Lopez, professor of microbiology at University of Almeria. And she will provide an overview about our project, and she will define the agri-food waste plastics. And finally, she will illustrate our solutions and innovations. So I'm sharing my screen now. Okay. Can you see it?
Yes.
Okay, so I play the video. Let's get started.
Hello, I'm Maria Jose Lopez, professor of the University of Almeria and coordinator of Recover, whose main aim is the development of innovative biotic symbiosis for plastic biodegradation and synthesis to solve the end-of-life challenges in the agriculture and food industries. In this presentation, I will give a general overview of the project and the consortium, and I will define also the agri-food waste plastic and the solution and innovation that Recover propose to deal with those plastics. The project started on the 1st of June 2020 and has a total duration of 4 years and a budget of 5.8 million euro. And as it is also for Encycle and Vicente, the basis is the application of microorganisms and enzymes to resolve end-of-life issues of plastic. In the case of Recover, we will use, together with enzymes and microorganisms, insects and earthworms, whose capability will be used to treat and bioconvert the plastic that are used in the food packaging and agriculture sectors. The consortium gathered the expertise of 17 partners from 7 European countries, including universities, associations, non-governmental organizations, small, medium, and large enterprise. The two sectors, food packaging and agriculture, consume around 44% of worldwide production of plastic, having many different applications. And after the end of life, the plastic that are collected separately, currently only 51% of this fraction is recycled, where the remaining fraction is incinerated or landfilled. This is because this fraction usually contain a mixture of different plastic or multilaminated material that are non-recyclable with the current procedures. And also there is a fraction of the plastic that contain the organic fraction that after composting remain in the compost, contaminating it, that then in turn will contaminate the soil when it is applied. Also, practices in agriculture, for example, the use of mulching films that degrade, break down in soil and produce microplastic, contaminate also the soil. Recover, we focus on this fraction of non-recyclable plastic and we try to provide new biorecycling routes. And also we will aim or target this to give a solution to the contamination of the compost and the soil plastic. And we will do so by combining the action of new enzymes, microorganisms, insects, and earthworms, whose biodegradation capabilities will be improved so the plastic can be removed from the compost and the soil, so provide environmental ecoservices. And in parallel, we will generate a set of products such as biofertilizers and chitin and chitosan that will be extracted by new innovative processes from the exoskeleton of the insect that transformer or reuse for the production of bioplastic or material are fine application in the food packaging and agriculture sector. So we close the loop by turning the agri-food waste plastic as a resource and meet the circular economy. During the development of the project, we will characterize the problem. We'll select and upscale the production of the biotech solution by combining them, and we'll prove the activity directly in soil, in situ trials, where we test the bioremediation capabilities to remove the plastic, ex situ in compost and vermicompost reactor and insect rearing chambers for the treatment, the conversion of the plastic to new added value products such as improved biofertilizer, chitin-based bioplastic and coatings. And the sustainability of the solution proposed will be also evaluated, as well as the impact in social, scientific, and economical point of view. This will be evaluated in two main scenarios that will be the food packaging industry and the agriculture sector. In the next presentation, the Recover partner will focus on the problem itself and the biotech solution, including the enzymes that we will develop in the frame of the project, and also the activity of the insects and earthworm for the biodegradation of the plastics. Thank you very much.
Okay, so as Maria said, the next part will be Marie Alimpirat from Idelux. She has a master in biological science, and she will be discussing about the sorting mistakes made by citizens and the consequences in the industrial waste treatment at the industry level. So let's see.
My name is Marie Alimpirat. I'm representing Idelux Environment, and I'm going to talk about the sorting mistakes made by the citizen. This environment is an intermunicipal, public and non-profit large company. Hires more than 200 people. We're managing household waste within the province of Luxembourg in Belgium. So here is the map with our capital, Brussels. And I draw a green light to show you our province. That's about 55 municipalities, and it's covered by 53 container parks where the citizen can sort their recyclable food chain waste, for example. So when mixed, it's considered as a waste, but once it's sorted, it becomes a resource, and that makes sense because if you sort by the type of polymer, for example, the food packaging waste then becomes definitely easier to enter the recycling procedure. I don't know how it looks like at home in your being. If you have only one bag and you mix everything, sometimes it's the case. Or if you have or if you ask like our citizens to sort your waste in two different fractions, in two different bags, or in only one bio bag, which is separated, then you have two different fractions, what's organic and what's not. Basically, it looks simple and quite easy. The reality looks more difficult for the people to sort their waste, because that's a picture I took from the fence. Some days it looks like that. So we have full of plastics, we have metal, we have rocks, we have glasses, and people just don't think about the treatment we have to do find. So they think it's just too difficult to sort the waste, not convenient, they have no space enough, they are not paid for doing our job, or they think that they pay a tax and so they don't have to do more, or they just believe that the wastes are mixed in the truck anyway. But in reality, for Idelux Environment, we have trucks that are very well compartmentalized. First, we have two different plants for two different purposes, one for the organic fraction and one for the residual fraction. That's completely different because the tools they have are different too, and they are geographically well separated. So it's a complete nonsense for us to have mixed waste. quite the opposite. So the residual fraction is prepared in Abbé as refuse derived through fraction for the incineration purpose. And in Tendus, we are preparing the organic fraction for entering an anaerobic digester, and then the digest state that comes from it is co-composted with green waste to produce a compost of quite good quality. And then the biogas from the anaerobic digester is used to produce heat and electricity. So that's the big procedure at our two plants. But since sometimes it's not well sorted by the citizen, some plastics enter the anaerobic digester, and then the digestate that comes out from it is contaminated. It enters the composting facility, and then you can imagine the difficulties we encounter with these contaminants to produce a compost of good quality. Also, there's probably quite a lot of microplastics that ends up then in the environment, specifically in the. Thanks to the pretreatment, we can remove quite a lot of plastics, food packaging plastics, and this goes to incineration. So that's a loss of money, a loss of time, and it's a bad life cycle assessment if you take into account the process, because it should have been sorted from the beginning and goes to some recycling routes. So that's too bad. Also, the composting facility, there's a lot of contaminants, and since we are shredding the green waste, and we're shredding and then we are sieving it, the pieces become smaller and smaller, and at the end it's impossible to remove it anymore, so we have to send some volumes to the collection center. So again, same story, it's a lot of money, lots of time, and like a kind of big process, then it's less good. So the opportunity for us to participate in such projects like Recover is to think to improve our compost at the end, because with the microorganisms and the enzymes, we could then make these microplastics disappear or at least be reduced. And if we think in the future, why not think about this process also for the other fractions that I just showed you before, because that goes to incineration, but incineration is just heat production. Here with the Recover project, we could think about having added value products like bioplastics based on kitting from the insect larvae, for example, or biofertilizers. So these products from this process could compete with the heat production from incineration, and in the future this could be of more interest also. So we are very happy to participate in the project of Recover. Thank you very much for you.
So in the next part, Ronan McCarthy from Brunel London University, he is working on the bioscience division, will be discussing about the plastic biodegradation by microbially derived enzymes. So in particular, how we will use bacterial enzymes and some result related to their use.
I'm going to talk to you about plastic biodegradation by enzymes. So a brief outline will talk about why would you use bacterial enzymes for plastic biodegradation. The discovery pipeline that we're using to identify novel enzymes in nature, some of the promising results that we've identified so far, and how we plan to create better nature enzymes for plastic degradation, and then talk about some of the next stages that we explore. So why would we use bacterial enzymes for plastic degradation? Well, over the last 10 to 20 years, there's been a range of exciting studies published that have identified a number of different bacterial species that are capable of degrading commercially relevant waste plastics into monomers that can be catalyzed downstream processes. So this suggests that Microorganisms and bacteria represent a very rich reservoir of novel functional enzymes that may be exploitable for plastic bioremediation. So our discovery pipeline is using a bioinformatic approach to screen, identify different enzymes that may potentially be capable of plastic degradation. We will feed these enzymes into a functional screen. They will be expressed in a heterologous host and their activity compared to a range of different plastics. Now, this discovery pipeline is fed by two different channels. The first channel aims
Rafael, sorry to interrupt. Can you please increase the audio of the video because it's really low?
Okay.
And yeah, increase it to the maximum, yeah, and maybe go back to start his presentation because this one was especially low. Thank you.
Okay, thank you, Marta. But can you see it right? The full
Yes,
screen?
yes, we can see it. It's fine. It's just it was really low, especially this one. Thank you.
My name is Ronan McCarthy from Brunel University, London. I'm going to talk to you about plastic biodegradation by microbially derived enzymes. So a brief outline will be that I will talk about why would you use bacterial enzymes for plastic biodegradation, the discovery pipeline that we're using to identify novel enzymes in nature, some of the promising results that we've identified so far, and how we plan to create better nature enzymes for plastic degradation, and then talk about some of the next stages that we are going to explore. So why would we use bacterial enzymes for plastic degradation? Well, over the last 10 to 20 years, there's been a range of exciting studies published that have identified a number of different bacterial species that are capable of degrading commercially relevant waste plastics into monomers that can be catabolized for growth or that can be fed into downstream processes. So this suggests that microorganisms and bacteria represent a very rich reservoir of novel functional enzymes that may be exploitable for plastic bioremediation. So our discovery pipeline is using a bioinformatic approach to screen or identify different enzymes that may potentially be capable of plastic degradation. We will feed these enzymes into a functional screen where they will be expressed in a heterologous host and their activity compared for a range of different plastics. Now, this discovery pipeline will be fed by two different channels. The first channel aims to screen environmental isolates and use comparative genomic analysis to identify new enzymes. So specifically what we're doing in this for this approach is to collect soil from plastic contaminated areas and also collect plastic pollution from different areas and isolate the bacteria that are growing within these soils or on these plastics. We will then spot these cultures on plastic containing agar plates and check if any of the microbes isolated are capable of degrading the plastic in the agar. Some of our promising results from this is that we've identified a range of different species of bacteria that when spotted on our plastic containing agar plates create large zones of clearance, suggesting that they're capable of degrading the plastic within the agar plates. One of the most promising candidates that we have is this one here that, as you can see on an agar plate containing plastic, over time creates a large zone of plastic clearance over 5 days. What we've also seen with this isolate is that if we check the supernatant or the spent culture media that this bacteria has grown in, we can see that the enzyme responsible for this degradative activity is also present within the supernatant, which is very exciting for downstream bioremediation purposes. We've also demonstrated that this specific bacteria is capable of growing in minimal media that has no other carbon source other than the plastic itself, suggesting that the bacteria can degrade the plastic and use it for growth. And as you can see here, it grows to quite a high optical density. Our next approach that we're taking with this with these selection hits is that we're using in-house next generation genome sequencing to sequence the isolates and do comparative genomics to identify the enzymes involved. We will also be doing transposon mutagenesis to potentially disrupt the enzyme or disrupt negative regulators of the enzyme to maximize its activity. The other arm of our biodiscovery pipeline will be using a synthetic biology approach to assemble different modules or protein domains that have potential enzymatic activity to create new chimeric proteins that may be capable of degrading plastic at higher rates than what is seen in traditionally identified enzymes. So for this approach, we've identified a range of different enzymes, including cutinases and lipases, that we've selected for further processing. What we mean by further processing is that we've synthesized these enzymes, modified the sequence for expression in E. coli, and we've also added a signal peptide. We've seen that a number of these enzymes lend themselves to bioremediation processes because they have very high levels of activity in a heterologous host, as can be seen here. And what we're now doing is assessing the levels of activity of these enzymes against a range of commercially relevant plastics to determine which plastics these specific enzymes are capable of degrading. We're also optimizing the activity of these enzymes by creating enzymatic chimeras, where we fuse domains of different enzymatic domains together to get dual activity, hopefully creating chimeric enzymes that are capable of degrading a range of different plastics. We're also doing directed evolution experiments where we're hoping to modify the sequence of the enzyme through directed evolution to optimize its activity. And we're also doing targeted and random mutagenesis of specific sites within the enzyme to maximize and optimize its activity. We also plan to do some byproduct profiling to examine the biochemical processes that these enzymes are functioning in, and to also examine the degradative products of these enzymes to explore if they could be integrated into any downstream processes. So thank you very much for listening to how we plan to exploit enzymes as a way of plastic bioremediation as part of the RECOVER project.
So in the next part, Arno Cordes from ASA Special Enzymes. He is a genetic engineer, and he will be discussing about the development of fermentation process and the downstream processing in RECOVER.
Dear project partners of Encycle, Vicente and RECOVER, I am pleased to have the opportunity to present one part of the enzyme development within the project RECOVER. At first, I will make some remarks to plastic degrading enzymes, which are known from the literature. Then, I will talk about the fermentation of enzyme-producing strains selected by the project partners Brunel University and University of Almería. Finally, differences between the purification of intra- and extracellular enzymes will be discussed. There is a lot of literature dealing with the degradation of different plastic compounds by enzymes and microorganisms. Enzymes mostly described are from the groups of lactases and peroxidases, manganese peroxidases, and esterases and cutinases. In this project, we will work with different plastic substances as LLDPE, which is linear low-density polyethylene, sold as films in the market, polystyrene, known mainly in the foamed form as styrofoam, and polyethylene terephthalate, used for the production of bottles. Although the chemical structures are quite different, you will find in papers that each of these enzymes was used for the cleavage of all three plastic compounds. Thus, all of these enzymes will be the subject of the characterization of the isolated microorganisms as well as of the fermentation developments. On the next slide, you will see the procedure for the development of the fermentation processes. ASA will receive native microbial strains from the University of Almería and recombinant strains generated by Brunel containing plastic degrading enzymes which were identified and optimized by random and site-directed mutagenesis. At first, the cultivation processes for the most efficient enzyme-producing strains are optimized in shake flask cultures, whereas the activities of the above-mentioned enzymes will be the analytical key parameters. In the next step, the fermentation processes will be established in lab-scale bioreactors of about 10 liters. After optimizing of the technical parameters as aeration, stirring rate, feed rate, etc., the processes will be scaled up into technical scale. In view of the enzyme costs, the purification process should consist of a minimum number of steps. The ideal case is shown on the right side of the figure. The microbial strain secretes the enzyme into the fermentation medium in large quantities. Here, the downstream process would contain only these steps: separation of biomass, desalting and concentration of the enzyme containing supernatant by ultrafiltration, and stabilization in order to get a storable product. If the enzyme is formed intracellularly, which is mostly the case for recombinant E. coli strains, this causes a problem. See left side of the picture. Because we have to destroy the cells by a high-pressure homogenizer or a glass bead meal, a lot of contaminating and undesired proteins as proteases are released together with the target enzymes. Consequently, we need 1 or 2 more purification steps before it is possible to ultrafiltrate the enzyme solution. Such steps could be ammonia sulfate precipitation and anion exchange chromatography. At last, I would like to thank you for your attention.
Okay, so last but not least, Professor Raúl Morales Herrero from Miguel Hernández University has a PhD in agrochemistry, will be discussing about the biodegradation process made by insect and earthworms, and in particular, how to use them for the plastic biodegradation.
Hello, my name is Raúl Morales from Miguel Hernández University in Spain. I'm going to talk about the degradation of plastics by insect and earthworms. The outline of our presentation include why the use of insect and earthworms in Recover project, how to use them in plastic biodegradation, something about the promising result we are obtaining right now, and also the how to enhance the biodegrading capacities we observed, and also the next stage of our project. The lignin is a perfect example of the possibility of establish a biocascade to produce a real depolymerization and degradation of refractant compounds mediated by macro and microorganisms. In our approach, we need to develop a cascade that promote the initial pretreatment of agriculture or urban wastes in order to facilitate the final biodegradation by microorganisms. In this way, we are going to use insect and earthworms to facilitate this process. As you know, there is increasing evidence about the activity of insects as Galleria or Tenebrio in chewing plastics, but can also this inset induce a real degradation related to the digestive activity of the intestinal microbiota? In other line, the earthworm have some specific activities on plastics. There is evidence about the biofilm formation on plastic surfaces in contact with casts and feces of earthworms, and also there's evidence about the microbiome adaptation and the production of enzymes in these relationships. For this reason, we are going to use insect and earthworm in biodegradation of plastics. First of all, by the selection of the specific insect that have proved capacities on degrading plastics, and also to develop some kind of pre-adaptation of these insects. In our collection, we count with Tenebria, with Hephaestia, with Galeria to do this job. In the land of earthworms, we are going to select Eisenia, Andrea Orfetida, and Lumbricus terrestris to work together our proposal in organic matter-rich media and in soil respectively. We developed some kind of microcosmos trial in order to conduct incubation in plastic feedstocks using insect and using earthworm in order to produce a response in the microbiome of this insect and earthworm, in order to monitor the adaptation to this environment, and also to produce some kind of response in terms of enzymes or bioindicators in order to be used in our profile, our scheme of biodegradation in using a microbiological cascade. We are monitoring the loss of weight of plastics. We are monitoring about the response of induced enzymes, the appearance of a specific microorganism in the insect and earthworms, in order to know how the esterase, lacase capacities exozymes are producing, and also to include these capacities in a consortium that include a combo of microorganisms and extracellular enzymes that can be able to depolymerize and degrade the monomers obtained by each kind plastics. So how to enhance the biodegradation capacities? Just to select, promote, isolate, and obtain a complex consortium of microorganism enzymes to be used also with the insect and earthworm in order to produce an enhanced capacities to biodegrade plastics. And our next stage, our next goals are to compare the use of this insect earthworm mediated degradative change with and without our microorganism consortia, joined together with our sector partners Nutriensect and Entogreen in order to validate our solution. Thank you.
Okay. So this is it for Recover. I would like to invite you all to follow us on our website, recoverbbi.eu, and on Twitter and LinkedIn and Facebook. So now that this is for Recover, it's time to move on with the Q&A section. Okay.
Thank you, Rafaela. Just 1 second. I would like to remind people to do the poll, answer the questions. There's still quite a few people who haven't done so. So while we have these questions and answer sessions, please, it just takes a minute to reply. Thank you. Rafaele, you are muted. I'm not sure if you are reading or not.
Yeah, I'm not reading.
So-
No, I'm answering the survey.
Ah, okay. All right, all right.
Okay. Mhm. Okay.
So, do you proceed with the? We have a question that while you reply, I might send this question.
Mhm.
Let me find it. I think it's for Encycle project. Patricia asked, how will you deal with copolymerized additives in PET, such as flame retardants?
Okay, thank you for this question. In that sense, the idea that we have in this project is trying to depolymerize the different polymers that appear in those packaging. And finally, we will implement new recovery systems, like, for instance, supercritical CO2 or another this kind of typologies, in order to recover specifically terephthalic acid or maybe ethylene glycol or this kind of monomers, in order to promote this repolymerization. And the rest of additive that may appear not only in the PET fraction but also in polyolefins and so on. The idea is to recover them and also evaluate their insertion in the value chain. Yes, of course, I have not commented in depth, but also the promotion of the purification and downstream processes technologies is also a opportunity in this project. I think that I assume that I answered your question.
Thank you, Licinio. I think Rafael.
Yeah, we have
Yeah, more. Okay,
more
perfect.
questions. Okay. Okay. This question is always for Encycles, and the question is that nowadays enzymatic recycling is a trend, but what is the new from Encycle in this field? This question can be for Linicio or Ashwin.
Okay, I think that in a general overview from this project, I think that we want, or we aim to cover the whole value chain, not only for enzymatic depolymerization of the different polymers, but also we are facing different technologies regarding the obtaining of new enzymes, as Christian commented before, regarding the direct mutagenesis, the generation of these new enzymes or the enzyme for these processes. But also, for instance, we are dealing with new methodologies for enzyme production. We are aware that the incorporation of enzymes in industry, this biocatalyzer has, are very expensive for implementation in the industrial processes, and for this reason we are covering different improvement in different stages of the use of enzymes. In that sense, I think that the general view or the general overview of the enzymatic technology for industrial application is maybe the biggest innovation of Encycle. In that sense, also Christian or Ashwin, if you want to add something.
I think it's okay what you said. Yeah, I don't have to add something.
No, I totally agree also with you, Licinio.
So next question is for Christian and from Encycle. And the question is, why do we find this plastics degrading enzymized nature? It is a molecular adaptation toward xenobiotic plastics, or just a side reaction that, for instance, PET is hydrolyzed by such enzymes?
Yeah, thank you for the question. This is a great question because, of course, we are thinking about whether it might be a molecular adaptation. However, the time since these xenobiotics are out in the environment to adapt it's very short. However, we know that these evolutionary adaptation can occur within a short time. However, now we are facing a complex polymer with a partially crystalline structure. So I strongly believe that this is just a side effect, because when we have a look on the enzymes, they evolved a long time ago, and they are attacking the natural polyesters, for instance, like cutin. And I think I'm quite sure it's just a side effect. However, we can now use that side effect and try to engineer the enzymes to make them even better and more specific towards the targets like PET, for instance. So I think it may take a long time until organisms or also microbial consortia will really efficiently show biodegradation of fossil fuel-based polymers. However, it is of course possible, I think. Yeah. So when we have a look on the on other natural polymers like cellulose, which are also hard to break down, so we see it happens in nature. However, I think it's just a side effect.
Thank you, Christian. Actually, there is another question for you. And what do you think? What are the maximum hydrolysis rates? How long will it take to actually degrade a post-consumer PET materials by enzymatic hydrolysis?
Yeah, so when we have a look on typical PET packaging, what we find in the supermarket, like food packaging or so, this is all the recycled PET. I think the thickness of the material is about 0.2 millimeters. So let's assume such a film thickness, and I think it's possible to degrade the whole film or degrade through the whole block within less than 1 day. It's absolutely possible. So also we see from the Carbios when they work with LCC, when you make the PET into a powder and you're increasing the surface drastically, you can also achieve reaction times of maybe 10 hours or so, maybe a half a day. However, for the complete film, I'm not so optimistic, but I think 12 to 24 hours is absolutely feasible for a complete intact film of about 0.2 to 0.3 millimeters. Yeah.
Okay, thank you so much. So if there are no further questions for Enzycol, we move to Vicente. So are there any other questions in the audience? I see here one question in the chat. I'd like to ask Aswin whether he thinks that the customers of companies like Indorama are willing to pay a higher price for green recycled PET packaging.
¿No te deja?
Here in the
Yes.
chat.
Okay.
Can you read the
Bueno,
question?
pues ya está.
Yes, this is Ashwin from Indorama.
Okay.
You can hear me, right?
Yes, we can.
Yes. I think regarding the aspect of customers paying a premium for green recycled PET packaging, I think there are two factors that you may consider. One is that a lot of our customers are now focusing more in the field of sustainability. So if you look at those sustainability reports, they're making claims that within a certain amount of years, they would like to achieve an X percentage of the total plastics that they use for their food products to be made out of recycled content. So that is one aspect. The other aspect is that within the European Union, we have this legislation coming up, which will, how do you say, enforce producers of plastics to make sure that the amount of recycled content will be increased. So I think those two factors will, yeah, how do you say, make us all together work to create a product that is one from an environmental point of view sustainable, but for the from an economical view also beneficial for the producers of such plastics.
Okay, thank you.
In that sense, sorry, I would like to add that I think that these technologies, not only for enzymatic recycling, but in cooperation with chemical and so on, could be complementary. I mean, if we have a pure stream of PET material, it's quite easy to recycle by mechanical recycling. But in case of mixed fraction and so on, I think this is the key point in which we could increase the recycling rates, and trying to be fully circular, as we said in his presentation. I think this is only to one a few words, additional words to that.
Okay, thank you. So, any other questions for Encycles? Okay, we can move on to Vicente. So there is a question for Ivo Enzymai. I guess I said it correctly. And the question is, which characteristics of enzymes can be modified by directed evolution?
Hello, can you hear me? I think, yeah.
Yes, we can.
Right? Yes. So what we are focusing in Vicente about developing these characteristics of enzymes are basically the activities that are not natural, of course, with these complex plastics. So yeah, we can improve the enzymes in many ways that can be interesting for the project, like also the stability at different temperatures for the process. But we are focusing mostly on the activity on these plastics. For that, we are thinking about the different chemical nature of these resins, and especially in the bonds and the chemical bonds that can be degradable. So yeah, that's what we are focusing right now in the evolution.
Okay, thank you. So next question is for specific polymers. So do you have the same chemical strategies to bring reversibility for all the reference resins that you mentioned? For instance, explosive vinyl esters, unsaturated polyester, and so
on. Yes, this is Alain Graillot from Specific Polymers. Thanks for the question. So in fact, the main difference between these reference resin are the specification for the targeted application. And so depending on the application, for instance, for the implementation processes, you have to tune the viscosity of the resin, for instance, or to tune the resin composition in order to reach the targeted specification. So even if the global approach is to insert Vitrimer technology reversible bonds in all the materials you mentioned, we cannot apply exactly the same strategy and use exactly the same building blocks in all this resin, because they are not fulfilling all the specification of the implementation process or the end users. So basically, we have, I would say, 4 or 5 different kind of Vitrimer or Vitrimer-like strategies: disulfide metathesis, transesterification, transamidification, amines. And this strategy can be implemented in these different resins and tuned in order to really respond to all the specification we identified.
Thank you. So another question is for Theo Delft. So do you think that the result obtained with the small model compounds can be transferred to the real substrate? The question is for you, Delft.
Yeah, Thomas is online. He's probably the one. Frank did the presentation, but he had to leave. But I'm sure Thomas, who is working on it, can answer it.
Can you hear me now?
Yes.
Yes. Yeah, that's, I mean, referring to your question, that is basically the major point. but you have to start from somewhere. So I mean, we first have to understand the enzymes, what they are doing, and how they are attacking the polymers. Also, with regard to AVO enzymes, to approach them, what kind of directed evolution approach they were doing. Because if we don't have any knowledge about where the enzymes attack, how can we then degrade this more complex thermoset resins?
Okay, thank you, Thomas. Are there any further questions for Vicente? Okay, if any, we can move on to recover. So I'd like to ask to Marie-Aline Pierart from Idelux. And you said that although the pre-sorting, all the waste treatment in your plants, the risks of having microplastics still exist. So in your opinion, what is the percentage for having a correct recycling rates among citizens to be achieved in order to decrease the presence of microplastics in your plants? Is there any percentage?
Yeah, I can tell you that. I have no idea about the percentage. What I can tell you is the fraction we have now that goes out thanks to the pretreatment, and we send around 18% of contaminants, not only plastics, also other stuffs that goes for incineration. So the pretreatment removes already 18% of what enters in the organic plant, and there's more, unfortunately, because the pretreatment it's shredding and then sieving at 60 millimeters. So there's a lot of things that can still goes inside the anaerobic digester. And then after that, we again make some shredding and sieving with the composting facility, but there are still plastics in there. But at least we already remove 18% of contaminants in the beginning.
Thank you so much. So I would like to ask another question to Ronan from Brunel London University, and the question is: which singular or improved properties will have the enzymes with respect to the non-plastics degrading enzymes? So are there any particular properties pursued for enzymes, for instance, thermotolerance or release of specific monomers from plastics?
Yeah, absolutely. So we're looking at thermotolerance as a key factor that is important and that we're trying to optimize. And we're kind of filtered that into a pre-selection as well, because some of the enzymes that we're selecting are coming from natively thermotolerant bacteria. So bacteria that can naturally grow at very high temperatures. So hopefully that will downstream improve the selection of the enzymes. We're also looking at stability and also the range of activity. So how many different polymers can these one enzyme or can one of our chimeric enzymes is capable of degrading? So these are all things that we're trying to optimize during our selection process and optimization process.
Okay, thank you so much, Ronan. So one question is for Arno from Cordes. Arno Cordes. So in your opinion, Arno, what could be the main blockage for upscaling the enzyme production? Arno, are you there?
I think he is in the attendees. I don't know if Marta is able to.
Yeah, he's unmuted, and he's also got right to share his screen. So, and he's unmuted actually.
Can you? You have unmuted my microphone.
Yes, you are there.
Yeah, there's very much echo. I try to reduce my speaker.
Okay.
Maybe it's better. No.
Yeah, we can hear you.
Okay.
I repeat the question. So in your opinion, what could be the main blockage for upscaling the enzymes production?
The main process or the main problem?
The main blockage, the main issue. Maybe.
The main issue?
Yeah.
Yes, the main issue is the downstream processing from the gene recombinant E. coli strains, because I think that the most of the enzymes which will be generated in the project will come from E. coli, and the most of E. coli strains are forming the enzyme intracellularly, and therefore this will be the most problem to achieve or to target to a cost-effective process that consists of the disruption of the cells, E. coli strains, and the removal of the desired enzyme.
Okay, thank you so much, Arno. So one last question is for Raúl Moral from University of Hernandez. And the question is, are the earthworm feed with plastics?
Well, I understand that you are talking about what is the relationship between the earthworms and the ability of degrade plastics. Our goal is very challenging because right now we are, as agronomists, we have right now agricultural plastics in our soils. So we need not to be focused only in waste treatment facilities, but also in the presence of plastics in our environment. So depending on the size of the plastic, the active surface of these plastics and the relationship with the extracellular or with the induction of some kind of external or stressing responses to our earthworms is the first goal to our view, in order to find the microorganisms and the enzymes to be provided to other partners. In addition, the earthworms, depending on the type, Lumbricus terrestris have a very active relationship with the natural depuring capacities in the soil. The feces and the mucus located or exudated by Lumbricus have a very strong capacity to degrade xenobiotics in soil, and probably we are observing right now with Chinese researchers and also researchers from Spain in Toledo that they have some specific capacity to promote, maybe not directly but indirectly, the degradation of plastic. So it's a very good question, because we are working right now in this topic, and what is the real behavior, because the earthworms, not all the earthworms eat or chew plastics. Maybe Lumbricus terrestris, yes, but not Eisenia. But in the next future, the composter of organic waste in European or all over the world treatment plants have the question: how much microplastics are in our raw organic material? It is possible to reduce this charge of plastics in our compost. It's going to be the European authority to establish some kind of threshold level about the presence of specific plastics in the compost. This could be a real problem if they match this figure. This is my question right now.
Okay, thank you, Raúl. So, are there any further questions from the audience? No.
I think that's all the questions. So I think we finished. I would like to thank you everyone who's joined us for this webinar. We all hope that the three projects, we hope you learn a bit more what we are doing, and if you have any questions, we've shared our emails, but you can contact us. You can contact me. I can redirect you wherever you want. We really thank you. We would like to thank Antonella as well as the project officer of Encycle and Recover, and also BBI representative. Thanks a lot, because we know how busy you are, and being here it's really appreciated. And well, let's hope we'll meet you in another webinar sometime, maybe soon, face to face. But in the meantime, contact us and let's keep in touch. I don't know if Licinio or Maria Jose would like to add anything else.
No additional words. I think that thank you all. Thank you, Marta and your team, for hosting this event. Great, everything on time. It's a very good organization. Thank you, the Recover team and entire Vicente team, and of course Antonella from BBI. I think that we are lucky to cooperate between all of us to boost this kind of technologies and to promote this kind of projects.
Well, nothing much more to add, because I think that this occasion to gather all the knowledge and put together what we are doing in the three projects to solve a common problem is a good time for all to spread what are we doing. Thank you, Itene. Thank you, IT, for the organization, and Antonella, of course, and all the audience for attending to this meeting. Thank you.
Well, and thank you, Rafael, as well, because he's been a really good host, and with all the questions and answers, you've done great. Well, see you all soon, I hope. Bye.
Bye-bye.
Thank you very much. Bye.
Bye.
Bye-bye.
A pleasure.